Thank you so much for having me here today and for selecting my talk. Today I'm going to talk to you about spinal control of copulatory behavior and sexual excitation in mice. The first thing I actually want to point is that this work was developed with Constance Lencho. She was a postdoc in the lab when we were doing this work, and she's now in Magdeburg with her own lab. So to start, I want to talk to you about the basic structure of sexual behavior in mice. And it starts with the male and the female together, and they have this appetitive phase where they are sniffing each other, namely doing this anogenital sniffing, and they are assessing if the other individual is fit to continue with the behavior. And once this is true, they will go to the consummatory phase of the behavior, where the male starts to do this mount attempt, where he's trying to grab the female by the flanks, and eventually he will be successful, and he will do these mounts with intermissions or thrustings that start actually with this probing, that is a very rapid movement of the pelvic floor that allows for the male to find the vagina of the female, and then he will do this rhythmic thrusting behavior. And this is how it looks like. First, a mount attempt, he tries to grab the female, and then eventually he will be successful in these mount attempts, and he will do these not rapid, but deep thrusting behavior, and eventually he will dismount the female. And this behavior is actually repeated several times, and after a variable number of these mounts with intermissions, the animal will reach a final mount where ejaculation will happen. And this is how ejaculation looks like in mice. It is this very rapid pelvic floor movement here, and it will fall to the side, and the behavior will stop here. And this really marks the change between this consummatory phase of the behavior to the refractory period, where the male can no longer engage in sexual behavior for several hours or even several days. And because of this striking difference when ejaculation happens, we were very interested in understanding how this is happening in the brain. So one of the information that we have is that ejaculation is supposed to be a reflex mediated by the spinal cord, namely the lumbar part of the spinal cord, that then is able to have a motor output to the pelvic floor, and it's also receiving sensory information, but it's also receiving constant descending inhibition from the brain that inhibits this ejaculatory reflex, and sending information to the brain about the sensory state of the animal and the internal state of the animal. And once this internal state is close to ejaculation, it will inhibit this inhibition from the brain, and the reflex can happen. Another important thing is that ejaculation is divided in two main points. The first one is the emission, that is the sperm that goes from the testes, goes through the seminal vesicles and the prostate, and is deposited in the prostatic urethra. And then the expulsion part, where the sperm is expelled from the prostatic urethra, and this is mainly done by a pelvic floor muscle that is this bulbospongiosus muscle that, by contracting, will expel the sperm. And interestingly, previous studies in mice show that if you excise this muscle, the animals can do sexual behavior quite normally, but they can no longer do ejaculation. And we were interested then in dissecting this circuit by using this muscle as our entry point into this circuit. So the first questions that we had were actually, where are these BSM motor neurons located, and what is their activity looks like, and in relation to the muscle activity as well. To answer the first question, we used fluorogold, which is a typical retrograde tracer that we injected in the muscle, and then it will travel to the cell bodies in the spinal cord, and this is how the signal looks like. So in blue, you have the fluorogold signal, in green, the Nissl, just to identify the segments, and when we do a reconstruction of this cell position, we see that they are in the ventral side of the spinal cord, namely in the medial part of this ventral side. And if we count the amount of cells per animal, we see that most of them are actually located in the lumbar segment 6, also a bit in S1 and L5. And this is the main hub of BSM motor neurons in mice. So now that we know where they are located, we wanted to know what is the relationship between their activity and the activity in the muscle itself. And to do this, we injected pups with an AAV with channelrhodopsin in the muscle that would retrogradely travel to the motor neurons again, and then we allow them to grow We allow them to grow to 2 months, and then we do EMG recordings in the muscle while stimulating optogenetically the motor neurons in the spinal cord. And this is the type of signal that we have. So this is the BSM tracing, the bulbous spongiosum muscle. And you can see that every time we shine the light, we see this beautiful burst of activity in the muscle. And this is just a control in the leg that you see that there is no muscle activity. If we quantify this, we see that most of the activity that we observe in the muscle comes when we stimulate above the lumbar segment 6 segment when compared to other segments of the spinal cord, which makes sense because it's where we have a higher amount of motor neurons. Also, it is interesting to see that they follow quite well the stimulation until 10 Hertz, and then it decreases the fidelity. So now that we know where these motor neurons are located, we were interested in knowing what are the presynaptic partners that are controlling these cells. And we want to know, namely, their location and possible markers to then use, and what is which ones are connected monosynaptically to our BSM motor neurons. To answer the first question, we used PRV, which is a pseudorabies virus that we injected again in the muscle, and it will travel transsynaptically, retrogradely through these motor neurons. So it will basically infect these cell bodies and then jump to the presynaptic partners, and it will continue to jump. And here we see the location of the BSM motor neurons in pink. And if we look carefully then into the spinal cord, we saw this cluster of cells that are around the central canal in the lumbar segments 2 and 3 that were previously also discovered in rats to be involved in ejaculation and the central pattern generator for ejaculation. So we were curious about these cells, and we also did a staining then for galanin because it was one of the markers that was previously identified, and we saw that there was a big overlap between the galanin signal and our PRV cells. So we decided to try to use these galanin-Cre animals that express this Cre in all galanin-positive cells, and we cross them with the tomato, and this way we can have access to our galanin-positive cells. And if we do an immunohistochemistry, you see that, again, around the central canal, we have a big cluster of cells that is galanin-positive. And if we count, actually, the number of galanin-T tomato-positive cells, we see that there is a cluster of cells that is mainly on the lumbar segments 2 and 3, which we believe are the cells that are connecting with our BSM motor neurons. So, however, this technique does not allow us to know if this is a monosynaptic connection because this virus keeps on jumping. It's not just one connection that it jumps. So we wanted to know more about this connection. So we used these galanin-T tomato animals, and we injected them with a Cre-dependent virus that expresses synaptophysin tagged with GFP. And this way we can see the terminals of these galanin-T tomato cells, and if they are close to our BSM motor neurons, that we identify by injecting, again, fluorogold in the BSM of these animals. And this is the type of signal that we are seeing here. So basically, in blue, you have the BSM motor neuron, and then in green, you sadly cannot see very well, but well, there's green buttons here that are connecting with our fluorogold motor neuron. And if we quantify the overlap between the fluorogold positive cells and our synaptophysin buttons, we see that most of our fluorogold cells are indeed connecting with this galanin-T tomato. So it seems that it is a monosynaptic connection, but we wanted to know if it really is physiologically. So we did also patch clamp recordings in these cells. So basically we used pups that express channel rhodopsin in all galanin-positive cells. We injected them with color toxin B in the BSM so that we can tag the neurons that we want then to do recordings from. And by doing recordings in our CTB-positive neurons while shining the light in the galanin channel rhodopsin cells, then we can see if this connection is monosynaptic. We also did recordings in CTB-negative cells just to confirm that this is true for our BSM motor neurons only. And this is what we observed. So basically, in 18 out of 21 CTB-positive neurons, we add these beautiful action potentials when we shine the light. So they seem to be connected with our galanin-positive cells. And in the CTB-negative, only one responded. So most of them are this type of response to the light. So they were not really connected. And we have further pharmacological experiments that prove that this is a monosynaptic connection. So now that we know that they are monosynatically connected, I'm just going to do a brief summary. So we know that the majority of the BSM motor neurons are in the L6 and S1 segments. There is a causal relationship between the activity of these motor neurons and the activity of the muscle. And we have this cluster of galanin-positive cells that are directly connected to the BSM motor neurons. So now that we know where these cells are, we wanted to know more about their role. So we want to know if we can elicit activity also in the BSM when we stimulate these cells, if they are specific for ejaculation, and what happens when we delete these cells from the circuit. To answer the first question, we actually used electrical stimulation. So we used adults where we stimulated electrically around the central canal the position of the galanin-positive cells while we were doing EMG recordings of the muscle. And this is the example trace of one of the animals. We can see here that the BSM, when we stimulate the first time, have this beautiful burst of activity in response. Again, this is a control from the leg. And then from studies in the rat, we know that these cells usually promote several bursts of activity in the BSM if you do several rounds of electrical stimulation. So we decided to see if this was true also in mice. But when we stimulated the second and the third time with electrical stimulation in the spinal cord, we saw that this signal was gone from the muscle. So it seems that somehow with repeated stimulation, you no longer have BSM signal. So, and this is just a quantification of this. So both the amplitude and the duration of the signal goes down with repeated stimulation. And we also confirmed this with optogenetic simulation using, again, the galanin chanorodopsid animals that I showed before. So we were puzzled by this since we didn't know why this was happening. And to confirm that everything was well with our stimulation protocol, we decided to try the same protocol that the rat studies show in our own rig. So we did electrical stimulation in rats, and we were able to reproduce the previous results in rats that with continuous stimulation, we always have BSM responses, so it doesn't seem that there's nothing wrong with our electrical stimulation protocol. So then we also wonder maybe it's something with our preparation. Maybe we are taking too long to do the first electrical stimulation, so we decided to wait 1 hour before we did even the first electrical stimulation. And again, we saw the same type of response. So even after 1 hour, the first one would have a big burst of activity that then would go away with the second and third electrical stimulation. So it doesn't seem to be anything wrong with our setup. So then we looked at the behavior and possible physiological explanations for these. And one striking thing is that mouse only have one ejaculation per session before they enter in this post-ejaculatory refractory period of several days, whereas rats have several ejaculations, even 5 or 7 ejaculations before they actually enter in this post-ejaculatory refractory period. So it may be that these animals indeed are in this post-ejaculatory refractory period when we do the several stimulations, electrical stimulations. So we decided to have the animals have an ejaculation before we put them down for the electrical stimulation, and also to have just sexual behavior in order to see if this would be set by the behavior that the animal was having before. And indeed, when the animals had an ejaculation before we did the electrical stimulation, we saw that the activity was gone even in the first electrical stimulation that we performed. And this was not true if the animal was just doing 5 months intermission, so they were not in this post-ejaculatory inhibitory period. And this is a quantification, so again, the amplitude, duration, and frequency of the signal is basically much smaller or even gone when the animal ejaculated before. So it seems that there is indeed already an inhibitory signal in the spinal cord when the animal ejaculates. So we wanted to know if these cells now are active upon ejaculation, since we observed these. And to do this, we used CFOS, an immediate gene that is a proxy for neuronal activity, and we divided the animals in 4 groups. A first group where the animals were alone in the cage, a second group where they were sexually aroused by having 10 minutes of interaction with a female, but not having other components of sexual behavior. Another one where the males were allowed to do 5 months with intermissions, but not ejaculate. And the final group where they were allowed to do the full repertoire of sexual behavior ending in ejaculation. And this is the type of signal that we observed. In pink here, you have the gallon-T tomato again. In green, the CFOS signal, and then the overlap. And I hope you can see that already just by eye. The ejaculation group seems to have a higher overlap between the CFOS activity. And when we quantify this overlap, it is true that the animals that had an ejaculation before have a higher activation of these gallon-T tomato cells compared to the other groups, but already the aroused and 5 mounts have a higher activation than just the cage alone. So it seems that there is an integration of the internal state of the animal already at the level of the spinal cord when he's with the female. So these prompt us to actually understand what happens if we ablate these gallon-T tomato cells. And to do this, we use a technique based on diphtheria toxin, DTR. We injected a virus in the gallon-T tomato animals in the lumbar segments that expresses a diphtheria toxin receptor. And once you inject then the toxin, these cells would be killed. And this is how we did the experiment. So we basically trained the animals once to see if they would do sexual behavior. We injected them with a virus. We waited 2 weeks for the virus to express. We trained them a second time to understand that everything was right after the surgery, the spinal surgery. We injected with the toxin in this session. We waited 1 week for the cells to be killed. And then we did until 3 days of experimental days. So basically, in the first day, if the animal would already show any sexual behavior, we would stop the experiment here. If he wouldn't show any sexual behavior in the first day, we would repeat until 3 other sessions, 1 session per week. And we decided to do this like this because in our lab, we know that the animals are never longer than 3 weeks without doing any sexual behavior. And this is what I'm showing here. So I will show here an histogram of the behavior. In yellow, you will see the mounts with probing, which is the rapid movement to find the vagina of the female. In green, the mounts with probing and intermissions or thrusts. And then in pink, ejaculation. And each bar here is it's one of these mounts, and this is all the animals we observed. And in the sham animals, I hope that you can see that all of them ejaculated and quite fast. But when we look at our DTR animals, we see that already 3 of them did not ejaculate even though they started the behavior. And this is something that was never observed in our lab. Every time that the animal would start the behavior, it would always ejaculate, so this was very new. And besides this, most of the animals actually took more than 10 minutes, and when compared to the sham that took way less, this was also a difference. And when we quantify a little bit better this behavior, as expected, the latency to ejaculate is much higher in the DTR animals. And also this probing behavior, it's also altered. So the number of mounts with probing and the duration of the probing behavior is much higher in these DTR animals. However, when we then quantify more specific more specifications for the mounts with thrusting and for the intermissions, we see that this difference is gone. So basically, we think that there's an alternative path that once the animal starts to do this mounts with intermission, is activated, and the behavior is quite similar. So I hope that I convince you that we have these BSM motor neurons that receive synaptic input from galanin cells, that these cells are active during sexual behavior, but are more pronouncedly active with ejaculation, that the internal state of the animal seems to be integrated already at the level of the spinal cord, that the ejaculatory refractory period abolishes this galanin-positive mediated activation of the BSM, and this brings to the light that the ablation of this gallin population disrupts normal copulation in mice, which brings to the light that this spinal cord circuit is involved in more than just the ejaculatory reflex. And I just want to finish by thanking our lab, which is us here, mainly Susanna and Constance for all the help with the project throughout the years. Also Liliane and Bertrand for the help with the histology and analysis, our collaborators in France, and the platforms here at CCU, and you for listening.